Good evening. My name is Wesley Ray. I'm a student with Grand Canyon University, and I'm wrapping up my graduate program here with a class in biochemistry. Tonight's topic that we're going to be talking about is cell cycle regulation. It sort of falls in line with the topic of signal transduction pathways. They're deeply related. Um, I will apologize first and foremost. I've had a kind of a busy day. My voice is a little bit raspy, and it is currently 3 in the morning where I currently am. I'm going out of country in a couple days, and I wanted to kind of knock this out. So it's a little dark, and my wife's asleep, but we're going to get through it. So I'm going to go ahead and share screen. I don't want to drone on too awful long. Let's go share screen options. Oh, you know what? Let's just share to that right there. All right. With any luck, you're seeing what I'm seeing, and that should be my presentation on the cell cycle. So I wanted to go ahead and lay ourselves out some lesson objectives. Okay. We need to define the cell cycle and its steps. For most of us, I'm hoping that this is review. I think we cover cell cycle for the first time whenever you take your first high school biology class. So though admittedly it can be helpful to sort of go back through. We need to examine regulatory mechanisms of the cell cycle. Given that this is kind of the point of the presentation, we're going to be in that section for the longest, and that's why it's bolded and underlined. Okay, and then section 3, a look into oncogenesis as a model of importance. That's also bolded and underlined because we're going to be spending a little bit more time on that than what we will spend doing review on the cell cycle. Okay, but these are the 3 points that I'm looking to hit this evening. Okay, we're going to begin with the cell cycle. You have a diagram here of the cell cycle, a fairly simple one. There are 4 main steps that we've all been taught to the cell cycle. I'm listing them in order. The first one is the G1 phase, also called the GAP1 phase. This phase is dedicated to the cell essentially putting on weight. The cell is producing genes and it's producing proteins with the goals of growing. Um, if cells didn't grow and every time they reproduce, they split in half, daughter cells are half the size of parent cells, and sooner or later, cells would be so small they would be non-functioning. Growth is a direct requirement of the cell cycle. And so it shouldn't necessarily surprise folks when I tell them that the G1 phase is the longest of the phases. All three, the G1, S phase, and G2 phase, actually make up a kind of a combined lingo term called interphase, which is just everything except the actual actions of mitosis. The S, or synthesis. Oh, sorry. Like I said, long day. The synthesis phase. This is the phase where we begin to copy all of our DNA. There's kind of an important step that happens between the G1 phase and the synthesis phase. But in the synthesis phase, we are dedicated towards reproduction. We're going to go through the reproductive cycle for the cell, the mitotic cycle. And we're cloning all of our genes in order to make that happen. In the G2 phase, this is sort of a final prep phase. It's an extra thing with these eukaryotic cells. Um, Essentially, you're finishing off with proteins for growth, and you're laying the framework for the proteins required to perform the actions of mitosis. Okay. And then mitosis itself is the cell's mode of reproduction. Okay. It's essentially binary fission. Now, I have a second diagram that I've included below that shows very similar information with some cute little pictures of cells attached, but it has this extra R value. And that's called the restriction point. We're going to come to this in a little bit more detail later, but it's essentially a point of no return where the cell decides what it wants to do with its life. Okay? That's really all I want to discuss in the terms of the cell cycle and its steps. Like I said, it's going to be fairly quick. Most of this is going to be in the regulatory mechanisms of the cell cycle. Okay. So going from G1 to the S phases. Okay. This is the phase of tightest control in the cell. This is where the cell is sort of off and running. It has been born through the process of mitosis and it is entering into G1 and developing for the first time. Genes are being copied for the first time. They're being used and they're constantly being audited. Okay. So this process of G1 control sort of centers around CDK activity. These are cyclin-dependent kinases. Okay. Cyclin is a kind of a type of growth factor, and a kinase is any of these protein enzymes that act by phosphorylating other proteins or structures. Okay. So this is a cyclin-dependent phosphorylating enzyme. Okay. It's a serine threonine kinase, and it's activated by separate cyclin proteins. These start as separate subunits and they kind of combine to form this larger complex. Cyclin production is cycle dependent, so there are different types of cyclins that are produced that help to kind of guide the cell through the different phases. GAP1, S phase, GAP2, and even through mitosis itself. Okay. Kinases are kind of produced according to a positive feedback loop. The more we make of them, the more we make of them, assuming that they're being used correctly and assuming that there's not other problems going on with the cell. Okay. Um, CDKs, cyclin-dependent kinases, are going to promote E2F protein production. I'll get into what E2Fs are in a little bit more detail here in a minute, but they're basically their transcription factors for growth and development. Okay, so moving on. I'm not going to get into the diagram down below in a lot of detail yet. I included it in there because it's very nifty that it includes the phosphate group, and I thought that was relevant. Okay. Also, the chart up top shows you how the different concentrations of the different cyclin proteins will vary uh, as we go through the cell cycle. So not all cyclins are created equal. Okay. So the G1 to the S phases continued kind of to build on that a little bit. CDK proteins, cyclin-dependent kinases, they're pressuring cells to committing to advance through G1. And they're doing this by sort of encouraging the production of the E2F family of proteins. These are transcription factors that promote the production of genes that are vital to cell growth and reproduction. These could be anything. We could be talking about different cyclin proteins. We could be talking about DNA helicases and polymerases. I mean, you're talking about the things that cells need in order to keep going with their lives. Okay. They are sort of inhibited by pocket proteins. So if you ever hear the term pocket proteins being discussed, and there are a lot of different types of them, I have not named all of them here, but long story short, everything here's got a check and balance, and these E2Fs also have checks and balances that can be generically referred to as pocket proteins. One of the interesting things is that pocket proteins are being looked at as possible therapeutic agents because E2F proteins a lot of times are sort of agents that are related to cancer development when they are mutated and have gone uncorrected. So if you can inhibit the synthesis of an oncogene, uh, then you sort of stall cancer's growth and development, or at least slow it down. That's the theory. So there are a lot of people out there trying to sort of synthesize new pocket proteins with the goals of trying to slow cancer growth, or potentially stop it altogether. E2F transcriptional factors can be associated with growth or cell death, depending on which transcription factors are produced. There are different E2Fs to produce different transcription factors. Some of them promote growth and development. Some of them promote not growth and development. Some of them are kind of leading to the apoptotic pathway, things like ARF and P73. Okay. The cyclin proteins, the CDC proteins, those are growth factors. Those are useful to the cells. We talk about cyclins a little bit more as we go on, and I've seen ARF pop up a lot in literature. The restriction point. Told you we'd come back to it. This is a point in the G1 cell cycle phase where the very dynamic pressures encouraging and discouraging cell cycle progression result in an all or nothing commitment or pause. So you've got a lot of different pressures from growth factors that are encouraging cell growth and development. You may have some death factors. You may have some factors that are there trying to stall growth and development. And essentially, it's a tug of war to see which one wins. And once we hit the restrictive value, it's sort of this point in the cell cycle where if the death factors are not there in a sufficient enough amount, the growth factors are about to become self-sustaining. The CDKs, the cyclin-dependent kinases, and their actions are going to result in a positive feedback loop that will keep the cell moving through this cycle once we hit past this point. So this is the point of no return. The cell is either going to go into apoptosis, is going to not develop any longer. If it doesn't develop any longer, it's said to go into a G0 phase, a gap zero phase, which is not included in the typical model of the cell cycle because it's not ideal, but something to keep in mind. Okay, so we've got a whole gradient of forces here pushing for growth and proliferation or pause and die. But at the end of this restrictive point, it's like a light switch. We're either on or we're off. So G1 cyclin production is positively driving the cell through this restriction point. G1 cyclin, which was the first of the cyclins that was shown in our cyclin expression cycle chart in the top right, G1 cyclin is being produced pretty heavily in the G1 phase. Okay. It's leading to a production of E2F factors that are encouraging further G1 cyclin production. So what's going on here is our CDKs are phosphorylating a sort of regulatory structure called RB. Okay. RB stops the production of E2F proteins. It's an inhibitor of them. By phosphorylating it and deactivating it, the E2Fs can be transcribed, and then they themselves can go to work creating all sorts of other different pro-growth proteins. So CDK complexes are leading to the inhibition of RB. Okay. By leading to the inhibition of RB, they are inadvertently promoting E2F transcription factors. Okay. The E2F transcription factors are themselves promoting cyclin proteins, and the cyclin proteins are themselves promoting CDK complexes because they're an active part of it. So more CDK complexes means less active RB, means more active E2F transcription factors, which increases growth and development. This is a positive feedback loop. The more E2F transcription factors we have, the more E2F transcription factors we're going to get. Sorry, I have a bird and it whistled, made a noise. Anywho, some of these things that we're getting out of the E2F transcription factors, some of them are relevant to the cell cycle's regulatory mechanisms, things like the cyclins that I've mentioned, cyclin E. Some of them are reproductive hardware. Again, we're not only trying to grow, but we're preparing to clone all of our DNA. So you're getting things like DNA polymerases out of this too. Okay. Keep in mind the whole mitotic process has got to be gone through in order for this to work. So reproductive hardware is being formed in alongside growth and development hardware. So at the restrictive point, let's make the assumption that we go through the restrictive point. The cell does not enter G0. The cell is going to continue on into S phase, later on into G2 if it is not arrested. Okay, so post-restrictive point. Cells are progressing regardless of environmental growth factor signaling. So they are no longer requiring external growth signals. Okay. They have enough intrinsic growth signal to proceed with their process unless something is going wrong with genes. Okay. It is stuck in DNA replication mode in a good way. That's a good start. Okay. G1 cyclins are going to result in the timely production of S cyclins and M cyclins. Again, the chart that I showed you earlier, G1 cyclins are going to be responsible for the transcription of some proteins and not the transcription of other proteins. Okay, we have different cyclins for different transcriptive factors. If all is well, the cell moves on to replicate, but it has to pass through some checkpoints along the way. Those checkpoints are important. They are regulatory mechanisms that would stop a, let's say, altered cell from going through the rest of this cycle and passing on potentially negative mutations and negative side effects. These cell cycle checkpoints. The cell's tightly monitored to ensure DNA quality, cell size, and overall positive progression. We're auditing for all sorts of things. We're auditing for DNA damage, double-strand breaks, and overall genetic instability, genomic instability, even, are all things that can sort of, I'm going to use this loosely, fail a cell's checkpoint. Okay? Failing a checkpoint means that you have got factors that are being expressed as a result of damage. That was like ARF I mentioned earlier. ARF is one of those that's being expressed as a result of damages. P53, checkpoint kinase 1, CHK1. These are all proteins that are sort of pausing and arresting cell cycle development, each in their own way. And some of them even helping to promote the transcription of cell death factors, things like caspases. Okay. P53 specifically is a very well discussed and very well characterized tumor suppressing transcription factor whose expression is triggered by post-translational modifications. We're talking about DNA damage. Okay. P53 can arrest CDK complexes. By arresting CDK complexes, we're allowing RB to do its job and prevent E2F transcriptional factors from doing their job. So we have sort of I'm deregulating the regulator on transcription factors, sort of a double negative. This is more present in higher organisms, things like eukaryotes. Okay, if you're looking at bacteria or some archaea, you're not going to find P53 expressed. It just evolutionarily it didn't show up at those times. Checkpoint kinase 1, it's a control kinase that can phosphorylate important promoters, WEE1, CDC25, and helping to stall cell progression until DNA damage is repaired. Working similarly to P53, just working on different target vectors. Okay. The goal here is not that we're permanently shutting down the cell. The goal initially is that we're sort of slowing cell progression or stopping cell progression until the point that DNA damage can be repaired. We need to be able to repair damages to DNA. If we cannot do that, then we may have to go down the death pathway. But if those fixes can be made, then fantastic. The cell would probably prefer not to die. So if a checkpoint is failed, there are essentially two main pathways in cells. Prolonged expression of things like P53, they'll result in cell death. They'll result in apoptosis. And there's two main ways here. The mitochondrial death pathway, wherein P53 enhances the expression of the BCL2 family of genes. This is occurring in mitochondrial membranes. Okay, they're subsequently released from the mitochondrial membranes and they trigger for caspase activation. That's a whole cascade where we begin to shut down all sorts of transcription factors and the cell begins to enter apoptosis. Or we have the cell receptor death pathway, which centers around receptors that are embedded on the internal side of a cell membrane. So they're inward facing. They're not facing outside of the cell. They're sort of facing into the cytoplasm, into the cytosol. and ligation of these different signal structures triggers for the promotion of caspase proteins, beginning a whole caspase protein cascade, resulting in cell death through apoptosis. Um, the goal here is not necessarily apoptosis, but if this signaling keeps going on and it doesn't cease, the end result is going to be apoptosis of the cell. Okay. So at this point, I feel satisfied that we've kind of talked about some regulatory mechanisms in the cell cycle. We've talked about growth and development. We've talked about arresting, and if the arrest is not temporary, then we've talked about cell death. Um, I mean, these are the things that are sort of regulating the cell cycle. If I was to go back to the slide discussing the restrictive point, there's really only two options. We're either going forward or we're not. So these are the two big that I've focused on as far as these regulatory mechanisms. And this third point, looking at oncogenesis as a model of importance. Okay. When you want to test the importance of a gene, it's really convenient to just look at what happens when it's not there. And I'm going to talk about an article that I'm very familiar with by doctors Hanahan and Weinberg. And this is the hallmarks of cancer. Now, they wrote this or it was published in 2000. And there was a next generation, Hallmarks of Cancer Next Generation, I believe, that came out in 2011 by the same authors. And these are some of the most cited articles on cancer on the entire internet. That's a big deal. These articles sort of changed our understanding of cancer and sort of fundamentalized it, if that makes sense. And a lot of the work and a lot of what they're describing has to do with signal transduction pathways because cancer arises when you disrupt them. And that means cancer is arising when we're disrupting cell regulatory mechanisms of the cell cycle. Okay? So I've included in here a couple diagrams from their article from 2000. And the one on the left is talking about the hallmarks as they outline them specifically. And I want to talk about the growth, self-sufficiency and growth signals and evading apoptosis. Those are the two biggest ones that I want to talk about right now. Now, there are explanations to the rest of them, but they're out of our scope right now. I don't want to, it's not the topic. On the right, I have a diagram of signal transduction pathways at large that sort of is modeled to try to explain what's going on with these hallmarks of cancer. I just want to point out to you that if you look at the top right with the anti-growth factors, if you're looking at the left side with growth factors, if you're looking at the death factors down in the bottom right, you'll see kind of where some of these players I've been talking about, BCLs, um, CDKs, cyclin proteins. You'll start to see where a lot of these are coming in. So I've included it there for relevance. It's a part of a bigger picture. Okay. Cell cycle regulators in cancer. So P53, I talked about that it was one of the most widely characterized of these sort of involved factors. And that's because it's nearly ubiquitous in human cancer cases. P53 is a tumor suppressor gene. It arrests the cell cycle when there is DNA damage. And there's DNA damage in cancer. Period. Cancer is arising because of mutations to regulatory mechanisms in cells. That's why they grow out of control. That's why they form tumors. And so yeah, P53 being mutated, very common in cancer cases because we're no longer regulating tumor production. We're no longer stopping cells from just going out of control. Okay. Uh, RB, I mentioned it earlier. It sort of represses E2Fs. If RB is malfunctioning, then you have uncontrolled transcription factors, and therefore you have uncontrolled growth and development. Okay, you have to have things in place to arrest the cell cycle when needed. And these arrests don't necessarily have to be like isolated incidents. They could be gradients. It can slow growth and development as a constant, like a constant rate overall through G1 and into S and into G2. That's not like a hard checkpoint point. but their influence helps to rein in the whole time growth and development for the cells. When you take these away, you get things like tumors. And when you have tumors, you're going to have metastasis. Uh, when you have uh, when you have cells that are growing out of control like this, it's the result of a lack of regulation. Okay, so you want to see the importance of these regulatory mechanisms? Take them away and see what happens. And what happens is cancer. I I I don't want to delve. I don't want to stay too long into oncogenesis and the models of its importance. It's a pretty simple point. But at this point, I feel like I've adequately discussed the cell cycle and its steps enough to sort of lay some groundwork into what I'm discussing in point two, examining regulatory mechanisms of the cell cycle. Again, I'm sticking with growth cycles versus or growth factors versus death factors there, more or less. I think those are really the two most important because we're either going forward or we're not. Um, and and looking at the importance of some of these in cases of cancer cell lines. Uh, so that's really all that I have to discuss this evening. I hope you enjoyed it, and if you have any questions, please do not feel free, or sorry, please do not be afraid. Please do feel free to contact me about them. Thank you very much. I'm going to go ahead and stop my screen sharing. Should be looking back at my beautiful face. I appreciate your time.